All right, everybody, we're going to be doing the second half of the chapter 14 notes on genetics, and this is going to be focusing on the statistical analysis or the math behind Punnett squares, as well as talking about your different forms of inheritance. So this slide right here is just talking to you about the idea of probability. So the first rule is talking about the rule of multiplication. So probability that two plus independent events will occur together in a specific combination. So in the case here, if we have a six-sided die and we have two of them, if I'm trying to roll a 6 on both dies, there's a 1 out of 6 chance that I'll get it on the first die, a 1 out of 6 chance on the second die. So if I multiply those two together, I'd get a 1 out of 36 chance of rolling two sixes on two dice. Another example is that if the chance of having five boys in a row, well, you have a half chance of getting a boy or a girl. So if you have a half chance of being the boy, and then another half chance of being a boy, then another boy, then another boy, and then another boy, the chances of having five boys in a row would be 1 out of 32, or 1, so 2 times 2 times 2 times 2 times 2, which gets you to your 32. And then this one right here was saying if you cross homozygous dominant, heterozygous, heterozygous with a heterozygous, 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 probability the offspring will be big heterozygous, heterozygous, homozygous, recessive is 1/2, 1/2, 1/4. Reason being is that there's only one opportunity of big C, little c, big C, little c, of it getting little c, so that's going to be that right there. The chance of getting big B, little b, if you cross a big B, little b, big B, little b, you would have two big B, little b's, so 2 of the 4 would be half, and then you have a big A, big A, big A, little a. The opportunity or chance of getting a big A, little a would be half, because half of them would be homozygous dominant, the other ones would be heterozygous, so half times a half times a quarter. So it's just giving you the idea of what the probability of getting this outcome would be. It's a quick way of being able to figure out, like, oh, if I want this specific outcome, just multiply it out like that, and you can figure that out. Rules of addition. So probability that 2 plus mutual exclusive ends will occur. Add together the individual probabilities. So chance of throwing a die that'll land on a 4 or 5. So it's saying that there is one chance of it having a fourth, right? You have 1 out of 6 chance of getting a 4, 1 out of 6 chance of getting a 5. You're going to add these options together, or these endings together. So a 6 and a 6 will give you a third. So it's a third chance on a 6-sided die to roll a 4 or a 5. So this is just helping you on being able to figure out other options that you could get. Like, what is my chance of getting a homozygous dominant or heterozygous? So you go back to these problems and you can kind of expand on the percentages of getting certain outcomes. Segregation of alleles and fertilizations as chance events. This is just telling you that it's just by chance that if you end up flipping a coin and you have a big R, little r on that side, big R, little r on that side, it's just by chance if you're going to get two big R's, two little r's, or a mix of the two. That's all it is. It's just by chance. So this is going to be focusing now on the other types of Punnett square questions, which we'll start off by talking about complete dominance. Complete dominance is what you're most familiar with, where you have a heterozygous and a homozygote. They will both show the dominant allele, and they'll be indistinguishable of each other. So if you had a capital Y, or a capital Y, little y, it doesn't matter. You're going to have a yellow seed. This was like when you have your big T, big T, little t, they're both going to end up being tall. That's complete dominance. The only time that I'm ever going to see the recessive allele is if you have two recessive alleles. So it'd be like little y would show up as a green seed, little t would show up as a small plant. That's how complete dominance works. There's a dominant, there's a recessive. The heterozygous will still show the dominant. Okay, now we have incomplete dominance. In the case of incomplete dominance, it's a little wonky. So instead of there being like a dominant or a recessive in incomplete dominance, we're just talking about dominant traits. So the idea is like allele 1 is dominant, allele 2 is dominant, and when they kind of collide with each other, they don't completely dominate over each other. They're incompletely dominate over each other. So what happens is that instead, the two colors are going to go ahead and mix. So in the case here, the hybrid, if I had a complete red plant, that's what this is over here. So I have C to the R, C to the R, and I got C to the W, C to the W. So RR is going to be talking about it being red, while WW is going to be it being pink. Sorry, it being white. So you have red here, white over here. If I have a C to the R that I got from this red flower, a C to the W that I got from this flower, if I combine them, CR, CW, I end up getting pink. So at that point, I'm going to go and create more gametes. So I have half of them C to the R, half to the CW. This is law of segregation right here. And I can set it up in this Punnett square right there to show that if I cross a pink and a pink, 25% will be red, 50% will be pink, and 25% of them will be white. This is incomplete dominance. This example right here is called, the flower is called snapdragons. They're a very common example to use in incomplete dominance, and it's a very by the book version of it. Okay, the next one we have is codominance. Now, codominance gets really confusing with incomplete dominance, and I understand the craziness of it. Codominance is that the phenotypes of both alleles will be seen. So, in the case of incomplete dominance, you had red over here and white over there, and they incompletely dominated over the other. In codominance, co meaning with dominance, they dominate with each other. So, in the case here, if you had red over here, white over there, and then they mix, well, it would still have splotches of red in some locations, splotches of white in other locations, and it would almost have kind of like a polka dot or like a mixed kind of look to it. Imagine like cats that have like mixtures of colors like all over their body. The orange, like black and white cats. The black and the orange aren't competing for each other, they're just seen in different locations, and then they're on top of the white locations. That's co-dominance. So the example that they have here is red hair and white hair would make roan horses. Roan meaning that it is R and W, but it's not actually pink. They are horses that have red and white components to them, red and white spots to them. Multiple alleles, this is one that I would maybe like add an asterisk to, because multiple alleles is an add-on rule for like codominance or incomplete dominance that's like located up back a slide. In the case of multiple alleles, it's what exactly it sounds like. Alleles is a version of a trait. So when we say multiple alleles, it means that we have more than just two versions of a trait. So instead of there being the dominant or recessive version of a trait, or there is the two different dominant versions like the red hair or the white hair, we're saying that there's more than two alleles. So think of this like if I had horns, right, on like goats. Well, some of the horns might be long, some of them might be short, some of them might be somewhere in the middle, some of them might have a little bit of a spiral to it, some of them might spiral in the opposite direction, some of them will be like ram horns where they're just like really like tightly packed with each other. Those are multiple alleles. There's more than two versions of the trait out there. The by the book example that we have here is human blood groups, where your A, B, and O blood groups, and this right here is just kind of showing you how this works. So in the case here, if you have the A allele, you will show up as having the A antigen on your blood. If you have the B allele, you will have the B antigen. And then if you have I, which is actually O blood, then you'll have no antigen. So this is what A blood would look like with all the A alleles all over it. Here's the B blood with all the B alleles over it. Here is the AB, because it's codominance and multiple alleles. We can see that the A's and the B's are mixed with each other, and then the O's, it's all by itself. This is just showing you the different phenotypes that are associated. Let's see if I can get back to it. The different phenotypes and the genotypes that are assigned to it. So in the case of A, it can be IA, or just be like AA. So two A alleles, or an A allele and an O allele, because O is actually recessive. So if you have an A and an O, or a B in the O, it's actually going to code for whatever the dominant allele is. So A has AA or AO. B blood has BB or BO. AB actually has one A allele and one B allele, while O blood will have this right here, two O's, or two little I's, recessive alleles. This is a little bit of a thing of showing you on what people can end up transfusing. So blood transfusion is very important. When you end up having foreign blood, you could have this thing called agglutination take place, which is blood clumping, which could cause to clots forming in arteries and veins, and ultimately leading to somebody dying. So we were able to find out about all these different groups where you have your O minus, O plus, B minus, B plus, A minus, A plus, AB minus, and AB plus. What this is telling you is who can end up getting the blood. Okay, so here's our donors, here's our recipients. O negative can donate blood to anybody and they'd be perfectly fine. Like they're set. In the case of B minus, they can donate their blood to any AB or any B. But if we go like to B plus, they can only donate to AB plus, or they can only donate to B plus. The idea on how this works is that, because we talked about the immune system, is that whatever antigens you have, whether it's the A antigen, the B antigen, A and B antigen, or no antigens, if you have the antigen, you have the opposite in antibodies. So if you have the A antigen, you have the B antibodies, which means that you'll attack anything that will have B blood in it. That's why A cannot donate to any of the B's right there. In the case of AB positive is the universal receiver, but the thing is that AB can because it has the A and the B antigen, it doesn't have any antibodies. So AB positive can like get their blood from like anybody, and it's kind of crazy how that works. If you have more questions, please ask. I only have about like a couple more minutes on this, so I need to go through the rest of the stuff. Here is polygenic inheritance, meaning there's more than 2 genes that inherit a single phenotype. The idea about how this works is that the more the allele you have, the more the expression will be. So in the case here, someone who has like all recessive alleles and all recessive alleles will have very, very pale skin. Sounds like a minus, right? It's exactly how that goes. Though if you have more dominant alleles and more dominant alleles, then it would end up having you having a darker skin color. So this is why when it comes down to skin color, there's usually like medians, or there's like a middle layer that you find a lot of skin tone to be, and then there's a variation of either getting darker or lighter from that. So the idea is that the more dominant alleles you have, the more of the trait you see. This is also how height works as well. This right here is talking about nature versus nurture. Genetic and environmental factors can influence phenotype. Depending on the acidity of soil, can actually change the color of these plants because it's turning certain genes on or turning certain genes off. Mendelian inheritance in humans. So this is by the use of pedigree charts. You're able to track to see if traits are dominant or recessive, and seeing how they're being tracked through here. So in the case here, this is probably a recessive trait because you can see that this individual doesn't have the trait, this person doesn't have the trait, but this one ends up showing up having it. So recessive, dominant, end up having a dominant kid, but probably they're heterozygous. This one's heterozygous and then shows back up there. Pedigrees are just a way of tracking a trait through a family. This is just an example of pedigree analysis. We can go over this a little bit more in class tomorrow, but just take a look at this. Just be familiar on how to read one of these, as well as trying to determine what kind of traits that we have. Your traits will fall under autosomal dominant, autosomal recessive, X dominant, X recessive, and then there's Y linked. And what is affected is just you're taking a look at who's affected, male or female, versus who is not affected. So just look over this, see if you can understand like all the different types of science right here, and then if you have any questions, just bring them in class tomorrow. Here's just a couple of different genetic disorders. So autosomal recessive ones, these are on your autosomal cells, so the non-X and Y cells. You can find cystic fibrosis, Tay-Sachs, sickle cell, and PKU right here, versus autosomal dominant. Huntington's disease is actually dominant, like it's a dominant trait. So if you have one allele of this, you're going to have Huntington's disease. The last couple of slides are just reviews of telling you about all the different types of all the different types of forms of inheritance. So just go through these and make sure that you don't have any questions. And if you have any questions, please feel free to ask me in class tomorrow, and I'd be happy to go over any of them. If you're unsure about epistasis, I'd be happy to tell you about that. And if we need to go into pleiotropy, we can go and talk about that as well. All right, thanks guys. Take it easy. Bye.